I find this meeting, it makes my skin crawl. It actually struck me of a poem that I love, and I won't do the whole thing, but there's a particular line. It's from Martha Graham, the line is that there is a queer, divine dissatisfaction. And I sit and I listen to all of this, and I want to get up and go do something because this is so big and so important and so challenging. And there is so much that can come together to make the world a better place. And it's the drive that gets up every morning. You should all read it. It's an amazing poem. It's titled Vitality. But I wanted today to sort of wrap this in a couple of brackets for the talk today. And this is a simple strategic planning diagram, which is where are we today and where do we want to get to in the exponential future? What's the current date from Ray these days? Is it 2027 or some point in time when the exponential integration occurs? And what I want to say, and I'm going to go through this a little bit, I want to go through a little bit of a real assessment of where we are today. And I want to talk about the fact that I don't think we can just leap to the exponential future, that we need to find some concrete ways in the middle to transition using these new technologies to make the world better, and propose some ways that we should focus our efforts to sort of begin to integrate things. So I'm going to start with my brother. And my brother Stephen was diagnosed with ALS in 1998. He and my family and I started the ALS Therapy Development Institute, which is now the largest and leading ALS research program in the world. It's a nonprofit. It has about a $10 million budget. And unlike most nonprofits, I didn't give any money away. I just started hiring people from biotechnology companies because I figured, why hire students? You want to work with professionals. And in this program, we did some interesting things, and I'm going to talk about a few of them right now. And in this photo, you see my brother Stephen and my daughter Zoe and a mouse with SOD1 ALS, which is a genetic form of ALS that's not in my family. ALS, by the way, is this motor neuron disease that robs you of your ability to speak and breathe, and it's very aggressive. It takes about 3 years on average. And Stephen Hawking is sort of one of the longest-lived cases that exists. You'll notice my daughter is not wearing a glove in the photo, by the way, in violation of the IACOC committee rules. But the reason I want to talk about mice is that we use mice, and in fact, much of the great research you've heard over the last couple days is about animal model research that allows us to understand how stem cells work, how drugs work, because we use those. Mice are essentially little supercomputers, molecular supercomputers that use these things called protein folding to accomplish the integration of biology in a way that real supercomputers don't do so well. And we use them to mimic biology. And they're models that are more or less appropriate for modeling human disease. And when you work on these models, you want to get a paper like this. And this came out early in my brother's illness, which essentially showed that Celebrex made mice live longer. And when you're an ALS patient and you're dying, this is a good paper, right? And we're going to talk for a minute about blue lines and red lines. And in general, in medicine, you want to be on the blue line. So in this case, the animals on the blue line lived longer, quite a bit longer, than the animals on the red line. And so immediately, Stephen started taking Celebrex with his doctor, as did hundreds and hundreds of other patients, or thousands across the world. And my lab, which was also testing mice, this wasn't my work, this is a friend of mine, Jeff Rothstein, who did this good work. My lab immediately tested this because we wanted to know whether it worked. And I wasn't actually looking to prove something didn't work, but I needed a positive control. When you do research, you need to know that your model's relevant. So you want something that works reliably in an animal and then works reliably in a human being so that you can relate the two. This is really important. So I tested Celebrex, assuming I would get the same result as this Hopkins researcher. That's really good work. And I figured, hey, I'm just this engineer. You guys are all the professional biologists. I must have done it wrong, so I'm stubborn. I did it a few more times. And I did it with mass spec, noting that the drug was at location and that we were modifying this pharmacodynamics, modifying the target. And my mice didn't live any longer. Now, in the meantime, they started a human trial. At this point, my brother stopped taking Celebrex. And I was struggling to make meaning out of this. And we worked on this for a little bit, and we asked the question, what happened here? So that's a graph of our control group animals from about 2004, 2003. It was about 1,800 of them. By the way, to give you a sense of how many animals this is, every academic in the world combined to that date had published on a total of 2,300 animals in the history of ALS. So I had a very big lab. And when I look at it this way, it doesn't look so good. We both buy our mice from the same place. They both have the same number of copies. I wonder if maybe the control group got sick rather than the treatment group got better. Because I don't think we would have put humans on a drug and the difference in the green and the blue line. Now, the trial happened, and in this clinical trial, that we had another line, and this was the answer. And in human beings, the blue line didn't match the red line. And there was lots of dialogue. Oh, the animal model's not relevant, it's not working for the disease. But we didn't really talk about what might have been the real issue, which is it may be that the model isn't the problem, maybe it is the way we use these models to do science that is the problem. So what we did is this is my lab from 2001 to 4, and you can see 3 different animal facilities. We were at Harvard at one point in our own lab, and from 2003 on, we were at the Mass College of Pharmacy for a little while. Tens of thousands of mice on this graph. And you'll see some animals that lost copies and lived longer, and some animals that we killed by accident by sticking needles in the wrong parts, and they died. And we used this huge database of animals to do something that had not been done before. So you remember that Celebrex result that I showed you? That's that little red dot. And what it is the number of animals, so somewhere between 24 and 30, and the effect, which is about 27% on the graph. And so that's the effect size and the number of animals. And that's Celebrex. This is every study published by everyone in the world as of 2005. And you can see, how much effect they observed and how many animals they used to do this. And we're going to do a little statistics here. So what we did is we built a Monte Carlo simulator out of our control group using the published parameters of every study published in the world, all 90, saying whether they censored, whether they did copy number verification, whether they balanced for gender, whether they balanced for weight, all variables that really affect whether the animals actually produce a real result. And somewhere between 50% and 80% of roughly $200 million worth of research in ALS could never have been significant. Could not have been. This is the only time anyone has ever done this in any field, reviewed and replicated the literature at that level. And I couldn't replicate almost anything. This is largely the drugs on the left that led to 7 human trials. On the right are my replications of the animal model studies that led to those trials. Again, with mass spec, pharmacodynamics. Roughly a couple $100 million in human trials resulted from this. 2 of the trials killed patients faster. And this is the best of science. I mean, ALS is one of the best research fields. And it made me really frickin' depressed. It was about 2005. Stephen was at that point in time really pretty paralyzed, and we asked ourselves a question. If we're going to live in the literature this way, how are we going to go forward? I mean, this is just, there's nothing truthful here to build the future on. And we actually said it will be cheaper to recreate the entire molecular knowledge of the disease than to separate fact from fiction in this field. And we tried to do that. They published all this work. What's interesting is that despite the scandal and the big press, nothing changed. And the criticism I got, well, the study design you're proposing is too expensive for any postdoc to do. No one can get the funding to do a study that will be significant. So we have to keep doing it the old way. And I'm like, this is not what we need. You can't have science that's driven by the needs of the postdocs. It has to be driven by the needs of the field to understand truth. And it's actually pretty easy to do. You could do meta-statistics on any model. I mean, it took us 15 years to know that the stroke models were not very useful. It took us a long time to know the EAE models were not useful. It took us years to discover that the sepsis model was not relevant. We could have just checked at the beginning. There's this thing called quality control that engineers do that biologists seem to don't know what it means. And I think this is a real challenge when we think about going forward into our exponential future, because at the moment when we do it the hard way, the old way, our results are largely bogus. So let's make sure in our exponential future we don't repeat the same mistakes of the past and produce a lot of false positives that mislead us. What's interesting is this is not just my results now. So this is data from Bayer on drugs that they bought. So this is stuff they licensed. Couldn't replicate 80% of the stuff they bought. So that's even after they looked at it and said, hey, this is a good idea. This is data from Amgen looking at replicating primary cancer findings that drove their program. 65% couldn't be replicated. Now, if you're not pissed, I don't know what to do. I mean, my brother died in 2006 on Thanksgiving. I didn't find a drug that saved him. 250 shots on goal. 250 drugs went into mice. Didn't find one that could make him live longer. So I did do some things, though. It's just I added this yesterday because Bob Hariri, who, by the way, where's Robin? I'm a hero of mine, too. He supported my research institute. He actually did this stem cell transplant. My brother is the second patient in this stem cell transplant that we did. And you can see here we did this back in 2001. And it took only 18 months to go from, I want to put stem cells in my brother, to actually putting them in my brother. Under IRB-approved conditions in the United States. We inject them as cisterna magna, as lumbar. And by the way, if you really want to learn how important medicine is, stand in an operating room when someone's about to put 100 million cells in your brother's brain and know that you're the one that made that all happen. It's very interesting. Take the responsibility of doing this correctly, really seriously. It's not, mistakes are very bad in medicine, and we have to really respect that as we move forward with technology that's going to shift everything, because the risks are real. Now, clinical research after discovery. So we just established that basically everything before clinical research is kind of largely bogus. Let's call it 70% roughly bogus. This is a clinical study that came out in PNAS that showed that lithium made patients live longer. So now we're not talking about mice. So now we're talking about patients. And of course, this is in 1998. My brother passed away, but hundreds of patients again started taking this drug. And a bunch of trials happened, 4 of them worldwide, to replicate that result. And again, the blue line didn't do any better when we replicated this. And this is about $30 million worth of follow-up research to refute this original positive finding in human beings. And again, it's not just ALS. John Ioannidis, who is probably the leader in this field, estimates that 80% of the clinical research can't be replicated. He just published this a little while ago. I mean, so when we sit here and we say, hey, let's just take all of the data in medicine and give it to people in better formats, or let's integrate all of the papers and give it to people and learn something. I think this is about separating wheat from chaff. We feed a lot of people a lot of straw that we think is wheat. And our job with exponential medicine is not to just create value out of this, the knowledge that exists, but to recreate it and prove what we don't know is wrong. Medicine is about doing less at the moment. So much of the potential we want is really just about, let's stop hurting people. I love this. She's the editor of the New England Journal of Medicine. It's no longer possible to believe much of the clinical research that's published. So now we've got discovery, and now we're going to go to clinical research, and now we're going to talk about healthcare. So this is a great paper, one of my favorites, and there's about 50 articles in the series. It's really interesting. They ask the question, according to the rules of clinical evidence, can a doctor tell a patient to use a parachute when they're leaping out of an airplane? Turns out, no. There are no double-blind, placebo-controlled studies that say that parachutes work. Anyone want to volunteer? Now, what's interesting about this, one of my favorite rebuttals to this was, no, that's actually not true. Originally, when they were designing the parachutes, they threw sheep out of airplanes with parachutes on them, and the ones that lived were the parachutes they used in the human beings. But we've already established you can't translate animal data to human data, right? So this is cute, because obviously you should use parachutes. Just as obviously, one should unblock a blocked artery in your heart if you detect it. Except you shouldn't. And I hate picking on the cardiologists because they are the only people that check. So the American College of Cardiology did a bunch of studies and registries, and they do a really good job. And they showed that if you are asymptomatic, if you didn't have angina, if you don't have angina, doing a bypass, all it will do is knock a few IQ points off you, take 6 months out of your life, and it won't have anything to do with whether you live any longer. We did 50 billion of those a year for a while. And again, I hate picking on the cardiologists because they actually checked and they stopped doing it. Except my dad was one of the last ones to get one. The problem is, and think about that, by the way, do we check on anything else? Do we do orthopedic checking? Do we check on gastric bypass? Do we check on any of these other things that we do all the time? We don't. We don't even know. So what's obvious is not obvious. We don't know. And even when we know the answer, I pulled this morning, actually. This is great. Dartmouth Atlas, you can just go and you can pull any kind of graph and look at anything. There was Bill just did a great presentation just a minute ago. But, this is just hip replacement rate, right? The number between 1.5 and 4.8, these are huge variations. Do we not know who this works for and who it doesn't work for? Do you know why? Does any hospital in America even know if anyone they gave a hip replacement to last year is even alive, let alone how well they could walk beforehand and how well they could walk, say, 3 months, 6 months, 5 years later? Do we know? No. We don't know. We don't even know if they're alive. So now, this is another great report from 1999 titled "The Air is Human." And I did is I put and it wasn't my idea, but I took this from another author who said, if the 98,000 estimated preventable medical errors, the things that killed people in hospitals, were put up against the CDC's cause of death index, it would be above diabetes. So not only do we not know if the hip replacement works, but if you come and get one, we're likely to kill you. This was replicated in September in the Journal of Patient Safety. The new estimate is 210,000 patients a year. So in the last 10 years, the estimates have changed, and it's gone up. In the United States now, that makes hospital errors above stroke the third leading cause of death in the United States. So, all right, let me get this straight. None of the evidence on which we base the clinical research that we do is all that viable. None of the clinical research we do is all that viable. We don't even follow the guidelines that wasn't that viable in the first place. And if you come and get the treatment that we're giving you, we're likely to really harm you. In fact, again, we're the third leading cause of death. I have personal experiences with each of these. Like, treated my brother with evidence that didn't work. My brother had hospital errors that almost killed him. The experience of this, it is going to require a fundamental revolution to change that. But the problem with this revolution is that it's not easy to get a better answer. We're thinking about how to do a new future. We have to do this in a new way. And Lord Kelvin, and my hero, and again, I guess he was the first quantified selfer. I don't know. But if you can't measure it, when you cannot express it in numbers, your knowledge is of a meager and unsatisfactory kind. And so I call this, look, people talk about evidence-based medicine. I was at the IOM about a year ago, and we were having this evidence-based medicine discussion, and I just kind of lost it in the meeting. And I said, guys, I'm tired of talking about evidence because there isn't any. And as an engineer, we have a fairly good rule of thumb. When you don't know what's going on, why don't you just start measuring what you're doing? So I think I coined the term, I'm not sure, I call it measurement-based medicine, which is, if you don't have any evidence, at least measure it. And I have this vision where there's three people in the room, right? There's the patient, there's the doctor, but there's a third person. There's this third patient that's just like the one there, it's a ghost. And they're going to come in the future. And if you don't write down what you did and what happened in the state to some degree that you can help that third person with what your decision that you made there, you've committed malpractice. Start treating 3 people. The one there and the one that will be there next. And that puts doctors in the information business. So how are we going to fix this? My way of thinking about this is we have to know what the hell health is. What is health, right? It's this cute little heart icon. Well, I'm an engineer. I love vectors. So let's start with a vector. What is my mobility, right? The evidence, the evidence is that mobility is actually probably the best indicator we have in our terrible indicators of how well you're going to do when you're old. So let's look at mobility. So this is lifespan. And here we go. That's my 3-year-old, my 5-year-old, my 12-year-old. I'm at 47, declining down here. My dad's at 75. Do we know this for anyone? It's a simple measure. How fast can you walk? How well can you move? We can measure this. It's not that hard. It'd be nice to know that, right? That's a simple one. What about something like well-being? How happy are you? I love Dean's talk. It just inspired me and made me, cry with the beauty of what he'd created and thinking about love and the human connection and the purpose of being and how you can create and empower that. Well, here's my well-being line. I was a very happy kid, born in London, enjoyed life, went to middle school. God, that was terrible. Went off, got to college. We were talking about dating Wellesley girls last night, and then, ended up, had children. Not so good. And then, I'm around here. So at some point in time, your children go away. They go back to college. And it turns out the evidence is, it's actually a pretty well-researched field, you almost get to the level of well-being you would have had if you never had them. By the way, there's a whole conversation here about linear scales and whether all points are equal and things like that. I think we have children for like that 5 minutes of joy each year, for which we suffer for the other 250 days. But eventually, illness starts to affect you, and you go downhill. And there's this complex relationship between you can't hear, or you can't see, you can't walk, and you become depressed, and it integrates into this catastrophic failure that Dean showed how you can address and adjust. We don't know this for anyone. In fact, I don't even think we've got a good measure. As was said yesterday, we need a measure of brain health. We need a measure of the human condition. I want a number that if I go from being, a 5 to a 6, I feel it. It means something. I know I'm better. I'm happier. Now, disease changes these things, right? So disease comes in. That's ALS, takes out your mobility, your well-being oscillates. It turns out ALS patients are actually pretty happy. I'm trying to figure out if there's something zen about just not being able to move that makes you sort of constantly meditate. But in that context, what's interesting is that every condition invents its own standard in which to be deviated from. Depression is different in cancer patients than it is in heart disease patients. And everyone's health state is measured. And as an engineer, it's like there was this thing, we had these guys that called blacksmiths, and every blacksmith had their own way of making things. And you couldn't buy something from one blacksmith and go to another blacksmith. Well, every disease is a blacksmith. They don't agree on what to measure. They don't agree on any common measures. There's no way to compare across them. Understanding how a treatment affects the mobility and the ability to live a healthy life of someone who is suffering from gerontology is different in congestive heart failure versus hip replacement. There's no comparability because there's no way to compare value. That's what has to change. And in order to think about that, I've been contemplating this problem. So there's another company I'm involved with that's got a health product, something that I think is going to really make people feel healthier. It's called AOBiome. And the challenge we have is I don't want to spend 16 years like Dean did trying to prove that this stuff actually makes a difference. I'd actually like to do it quicker. So here's a question. Let's imagine something for a second. Everyone here is going to spend the next 30 days, and here's what you're going to do. You're going to sleep every night comfortably and well under the right lighting conditions in silence and peace. You're going to eat spinach and fish and all kinds of healthy things for 30 days. You're going to exercise for 30 minutes a day, and you're going to meditate at some point in time on a beach staring at the horizon. Now, does anyone here not know they will be transformatively better in 30 days? How would I measure that in a clinical trial? How would I know? I mean, your cytokines might be higher because you're stressing your muscles. You're not going to move your lipids. So something that's so obvious, you can feel it. You feel that image? You can feel it. And yet we couldn't measure it. So if we couldn't measure it, how would we accomplish it? So this is what we have to work on. And this is the company that Ben and I and another friend, Jeff, built. And I'm just going to show you what it looks like. I'm going to search. I'm a multiple sclerosis patient. I come in, and you can see what it's like to have MS, the symptomology of it. You can see how people treat MS, brain fog. This is the patient's language, bowel problems, and how people manage those conditions, those subconditions. The treatments, how effective they are, how much side effects they are, how many people are in the system, what age they are. And you can click on these numbers and find the people. So if I click through to the 32,000 MS patients, you'll see here are the ones that were coming in. I did this last night, so you can see last night's updates. I can filter this to being, women. By the way, I invented this. I was a dating site. I was running a clinical trial, and I was online dating, and I'm like, peanut butter, chocolate. That was patients like me. And so I'm dating, so I'm going to look for a woman who's age 30 to 50, and then I'm going to filter it a little bit. I want someone that's had MS for a couple of years, so at least 5 years, but not so long, not since they were a young kid. So like me, I'm going to want someone that has, you see how some of these people are really red and some are yellow and some are healthy? That's because they all have different levels of disease, right? So I'm going to filter for how sick are you? So I'm going to take people out that are working. I'm going to go to a real level of disability, a 30 on the MSRS, a severity scale that you want to move up on. You want to be lower. And I'm going to pick someone between 30 and 50, and I'm going to come back and I'm going to find someone here and look for someone like me. So, click on one of the patients that I think is interesting and drill in. And what do we know about this human that shared her data to help me? So she's 46. She's in Altamont, New York. That is 4 years worth of data about the severity of her MS, her quality of life, social, mental, and physical, the relapses she's had, the drugs she uses to treat her disease against the indication. So Lexapro for anxiety and ibuprofen for pain and prednisone for the relapse and omega-3 acids to make myself healthier. The symptomologies of anxiety, stress, pain, sexual dysfunction. If you're an MS patient, these are things you want to solve, right? You want to look at this data and enrich it. This is the kind of data we could build to understand what really works or what does not work if we were able to operate this at scale. And you can click on something like anxiety and just click through. Hey, look, 70,000 people's current anxiety. What if I just want to talk to the severe ones? So I want to find someone else. And they have other conditions. They have, depression or ALS or other issues. How do they treat it? So let's look at Klonopin. 3,500 patients on it for anxiety and panic disorder and insomnia. But let's just look at, say, MS patients on Klonopin. So refilter that. 750 people, probably larger than the study that approved the drug in the first place, for a sub-indication. Side effects in MS, doses in MS, why they stopped using it, how long they used it, how much they spend, whether they take it all the time. All of this data is just shared by people that are committed to learning something. It's quantified self on scale, on rigorous measures. Every single clinical trial that this woman is eligible for, within 25 miles of her house, is told about her. And you just click, I only want to look at drugs, and I only want the ones in phase 3, and you can just find that data and connect it. So it's the opportunity to connect resources to patients in real time. This is the system that we're trying to build to understand disease. And you can see the trial. If you're the pharma company, you can click a button and recruit all 22,000 patients that are within 25 miles of your site. You can see the sites. So we do some things with this. We studied insomnia recently, and this is US data only. We had about twice this worldwide. And just to give you a sense, the National Sleep Foundation does a big global study about insomnia across the United States. 9% of Americans are at risk of insomnia. It's about 9% of this room. In our population, 76% were at risk of insomnia. Dramatically different. And what's interesting is that the specialists that treat these patients don't actually look at this very carefully. So if you can manage insomnia, which is potentially causally related to depression or diabetes, you need to understand these variabilities. And the things about a common platform is that you can look at things very specifically. What's the type of sleep issue each of these patients has? So falling asleep, awake during the night, waking too early, waking unrefreshed. What's the difference in the US and then patients like me. But what's also interesting is how do they manage it? Now, this is not, from the drug RX platform or something like that. You'll notice here a couple things that are interesting. First off, marijuana has 60% of the market share of Ambien as a sleep drug. And it's only legal in, 3 states? And what I found also interesting is that marijuana had a higher retention rate for people that tried it for insomnia and stayed on it for insomnia than Ambien, and certainly much higher than Lunesta or the other ones. So it's very interesting that you can get this sort of experience and look at this and see what's actually happening and connect. And this, this is data on thousands and thousands of people. It took us, by the way, 10 days to run this study. 10 days, just to get the data. I love this graph. This is a graph that says how much insomnia is in each disease. So you can see that fibromyalgia or chronic fatigue syndrome, there's a lot of insomnia. In RA, but there's a lot in diabetes, more than one would expect. ALS patients, maybe it's because their well-being is better, don't have as much. They're almost normal in that context. We just actually put this out today. We're looking at the relationship between stress and insomnia. We do patient returns. This is how we return the data to the patients. We make these nice infographics to make them digestible and understandable, to thank the people that did this. This is, insomnia may be one of the real primary drivers of the inability to work or be productive in disease. So maybe treating the insomnia is almost more important than treating the underlying condition to get people back to their lives. So what's the take-home of this? What we really need are patient-centered outcomes. We need to track the meaningful variables that impact an individual's health. Now, that's a very complicated statement because the meaningful variables to a newly diagnosed breast cancer patient or a 4-year ALS patient on a ventilator or someone that's been severely depressed for 3 months are all very different. So you need to really deeply understand the human condition and all of these things so that you know which ones are really meaningful to the individual. And you have to do it in a way that's human-centered. You have to design these variables that are clear and answerable and efficient and relevant and educational, harmless. It's really interesting. If we measured exercise the way we measured health, it would be like this. All right, you pathetic human being, how many push-ups less than 100 can you do? There's no aspiration in healthcare. I want to be well. I want to be fit. I want to be positive. So you have to make these measures aspirational to make this work. And they have to be actionable. Do they change? And these things are not things that we do. When researchers study disease, it's always their agenda. It's never the patient's agenda. So all the measures we use for understanding health, that I love, Beck, who invented the Beck Depression Scale. By the way, what is depression? Depression is what Beck wrote in one weekend 40 years ago, and he can't believe that everyone believes that depression is what he wrote one weekend ago, but it's what depression is. We don't have a fundamental understanding of what it really means, or the subtypes, the variants, or what we know about it. Internally, we call these things phenes. We're a phenomics company. And beyond that, what we're really thinking about are what are the phenodynamics? So how specific is your measure? Are you targeting, fatigue or energy-based depression, or are you targeting anxiety-based depression? And is it just like a drug? Is it hitting the target well? How accurate is it? Its range, its precision? What are the biases in the observation? What are the variations in the population, the dynamics, the relationships, this interdependence between things like insomnia and diabetes or depression? And at the end, what's the meaning of that variable in the human condition to each individual in the system? And this all comes together, in a system that sort of makes people better. And, it's interesting. I'm really a discovery guy. I want to find new treatments. I want to find new answers. And I sometimes dangerously say it's not to me about the patients. It's about the data they give us to change the way things work. But it turns out we actually do make them better. We published this. We're actually doing a study with the VA now, and the initial read of the trial, which we're going to publish, shows that we replicated this, which is that you can actually make the patients better by having them engage in their health. And what's really interesting is actually there's this great dose effect curve, which is the more friends you have, the better you are. And if you add this up, what's interesting, I just want to show you a moment here, because this is about, and there's audio in this. There's a woman in this video.
I just remember an overwhelming fear. I was terrified. A lot of doctors were giving very vague answers.
I really had no clue what was going on with me.
And I just didn't have anybody to talk to who knew what I was going through.
I was diagnosed with psoriasis.
I had Parkinson's disease.
Relapsing, remitting multiple sclerosis.
Refractory epilepsy.
When you find a site like Patients Like Me and you realize that you really aren't in this fight alone, it takes an awful lot of the fear away.
I was amazed at the wealth of information that I found on Patients Like Me.
The most valuable and helpful part of the site is the tools that I get to use on a daily basis to see and track my progress.
The most helpful part of Patients Like Me is the forum for me, because it's the place where the community goes to share what's going on with their lives.
It's like a whole group of friends that understand you more than anyone else.
There's a community out there waiting to help you. Somehow, somewhere on Patients Like Me is someone who is like you.
Being able to be a part of Patients Like Me and my data contributing to research is an amazing experience.
It's using information that's available and that we can provide to find a cure, and that's what we want.
I just love the chance to be able to help someone that has psoriasis like me live a little bit of a better life and not have to go through the same experiences I've had to go through.
Without Patients Like Me, I probably would have never found out about an epileptologist who was able to offer the surgery to alleviate the seizures altogether.
Patients Like Me has made a very big difference in my life as far as my medications.
Oh my gosh, since the surgery, life has been phenomenal.
Finding Patients Like Me was, quite frankly, the best thing that could ever have happened to me.
For the first time in 31 years, I have been seizure-free. I have been able to drive again, and all of that was because of the information that I gathered on the site.
We can do much better than fighting the disease as a group than we can as individuals.
People should join Patients Like Me because it is a great place to find answers to questions to resolve your health problem, whatever that may be.
On Patients Like Me, everybody understands. There's no judging. We're just like you.
You can get a lot of data, you can get companionship, you can get understanding, you get compassion.
You can talk to your family, you can talk to your friends, and they are going to empathize. But if you're talking to someone who shares your illness, they get it.
Yeah, I apologize a little bit for the commercial, but when she said she could drive, it's so challenging to figure out how to do this, because the things that make it work, is it about the social, or is it about the data, or is it about the integration? What is this new understanding of shared knowledge? I think this is stuff that we're just learning how to do. And I think that the hard part about this that I want to touch on next is we have a big task. I feel 5% done. I am so divinely dissatisfied with my website and how good it is and how many diseases we cover and how many people we fail. And I've got to figure out how to get there. And this is a really hard problem that's going to take a lot of work and, quite frankly, a lot of money. And so I'm going to talk for a second briefly about where we can leverage this middle gap, because this is not easy to do medicine. It's not cheap, and we're going to need to do it in a new way. So that's a raw diamond and a cut diamond and a diamond ring, and I put them up there because our client, while all of you might think, hey, it's the providers or the payers or all these people that want to make people better, turns out they actually don't. And I don't mean that they don't emotionally want to make people better, but they don't spend any money on measuring whether the people they treat get better. And if you define caring as measuring what you care about, they don't actually care. In fact, if you look at the whole world, doctors don't invest in measuring outcomes. Hospitals invest a couple $100 million in measuring the cleanness of the waiting rooms. Kaiser might be some exception to that. I'll give you some points. And then in addition, insurance companies don't measure things except whether you're going to your healthcare provider and HEDIS and a little bit. And then governments do some measurements, a couple $1 billion a year. The pharmaceutical spends $60 billion a year measuring whether things work. Now, why do I have the diamonds up there? If you're a pharma company, here's what you do. You go and you find something that might hit a target, And you don't know what it really is. It's raw, it's unformed, there's not much information around it. You cut it into the shape, which is finding the indication or the group that it will best fit with. And that process, is the part that spends the hundreds of millions or billions of dollars to produce something beautiful in the end, which is a molecule that can affect human health. And by the way, I've tried to do this. It's really, 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 really hard to do. And then they do this sales process where they dress it up in these kind of silly different rings and stuff, and they put one in Tiffany's, and they put one at Shreve Compton Low, and some at the mall, and you can tell about which companies sell at which price and which thing. But the real important part is the cutting of the diamond, figuring out how to make that diamond mean something. And I want to talk about these diamond makers for a minute. So this is global worldwide pharmaceutical sales for the last decade, and we're at about $856 billion a year. Billion. That's almost like Iraq war level spending here. So this is a lot of money. Interestingly, the U.S. sales went down last year for the first time ever by 1%. And if you look at this in another way, this is the number of chemical entities approved by the FDA since 1940. It's roughly about 25 a year. It's trending up. And I think about this, and I look at those two numbers, and I do a little bit of math. So there's $856 billion in global sales. There's roughly 65% of those come from branded or on-patent drugs, which means they have 10 years to live. You make the drug, and you get 10 years. It's about $550 billion a year of new inventions that modify human health, divided by 25 drugs per year. That means that each drug has a net present value, an expected value of $22 billion. And actually, it's actually roughly accurate. There's a company called Exponential that has one drug on market, and they're worth about $19 billion. And it's for a rare disease. So this is real. But there are 7,000 diseases. This is not going to work. And I think that we have to think about, now what I like about this graph, and it's really important, is that unlike the hospitals, which get to stay and they've got their political connections and they get to defend themselves, and the doctors that can maintain the Medicare reimbursement rates, and anyone that thinks the ACA Act is going to drive enough money to change the system in a significant way in the next 10 years has really not read the law. It's just not moving that much money. I mean, does anyone here change their behavior that significantly for a 1 or 2% income switch? I don't. But it's not that it's not worth doing. It's all worth doing. But these guys die. Every 10 years, they have to invent $550 billion a year of invention. They are the innovators. Now, there's lots you can criticize them for, but they are the innovators. They make movies. They have a half-life. The half-life is gone and the drug is gone. So they're the ones that are going to look at this exponential change and say, I need to embrace this or I'm going to get crushed. And if you want to see resources shift or change, move an industry that moves $500 million of invention a year, $500 billion of invention a year. This is serious, serious change. So how do we shift from where we are now to there? The answer is we do signal and stratification. Now, what is signal? We learn how to measure the human condition. The drug companies don't know how to do it. And you learn how to make me different from you, different from you, and different from you. What makes us all different? And we have to do this in a structured and effective way. And then we have to get the patients ready to be worked on by the drug companies. They're ready to go. The validated clinicians ready to go. There's a proven patient-centered outcome measures. There's known subtypes. And there's individual predictive models. And I want to touch on this real quickly. Many of you have seen our work in ALS, where I can look at this patient and say, computer, go and get me 10 controls for this individual. Tell me their future. So I can see whether the drug they're about to take is going to make a difference. So there are the 10 controls for this patient. It's going to make a prediction about his individual future on functional outcomes he cares about. And he died at the 75th percent of the controls exactly where expected. You could call a wheelchair, a death, a ventilator, any Kaplan-Meier data. This allows us to evaluate whether the drugs work in the real world in real time. This was the lithium data I told you about. We knew that drug didn't work before a single trial started because we measured the patients in the real world. It looks like this. You get a curve. That's how confident you are about the outcome. But this is what we've done next. There's 3 drugs in this. One is a chemical. It's called sodium chloride that people take. that made them worse. And this chemical, they believed it was helping them. We showed it was making patients 50% worse, and they stopped taking it. That's the blue line. The other two lines are really kind of cool. And as AJ, you still here? One of them is Biogen's clinical drug in ALS that they spent $100 million. We measured their trial using our measurement system and our data and called the result before they unblinded. NP001 is another trial. So we're calling drug trials, watching the real world. Now I have a question. How many drug trials do I have to call accurately? Because before someone asks the question about whether this is a useful tool for evaluating whether things work in the real world. Because I don't know the answer, and as far as I can tell, medicine, the drug companies care, isn't that interested in this problem. I'm going to personalize this, then I'm going to end this for a second. Remember my brother's stem cell transplant when I put the 100 million cells in his spinal cord? I never knew if it worked. Never knew if it worked. We built this prediction algorithm, and I get to ask the computer again, computer, tell me for my brother, 5 years after his death, whether the stem cell I gave him made any difference. So it's getting a bunch of patients. Notice they were all diagnosed years later, and it's comparing them to Stephen and making a prediction for Stephen. And the stem cell transplant, as we thought, didn't make any difference. And I had to wait to build this company to answer the question about the treatment I gave my brother in 2001. Think about what we can do if we make this global. We can change this. Maybe it's only 50 million to approve a drug, and you can make money on 100 million a year instead of $1 billion a year. We have to untangle this puzzle for the drug companies. They will give us the resources if we work with them as exponentialists to figure out how to understand the impact of health and the way Dean described it, disease and variables as all the amazing research you saw described it. And I think this last one's really important. What does it mean to grow old and die? What is longevity? What is the relationship between the molecular age of your body and your health and your diseases into this concept called health span, which is a word I love dearly, and I wish I knew who invented it. But it's how can I have the longest, healthiest life possible? And if we do this right, the exponential short term is that we go from disease to health, and we give ourselves immensely long health spans. And I get to have a second crisis here when I'm in my 80s, and we go on from there. So thank you.